Uh, Hi everyone. Uh, I guess I'll start now. So today I'll be discussing what has happened during week 4 and some of the questions related to that. Uh, just share the screen. Can you see my screen? Hmm, I guess yes. Okay. So, uh, so this week, sir has discussed some of the basics of microscopy, and uh, he has sort of ventured into what the structure of cell looks like. Uh, so coming to the basics of microscopy, you so this is one of the first microscopes that was made, which by Zacharias Janssen. This particular microscope, it's super old. It dates back to 16th century, but it could only go to 3x magnification. So the ob- object you are trying to image, you can only zoom it up by 3x. Uh, there were subsequent approaches to design better microscopes, and one of the first simple microscopes that sort of helped us set path for microscopy in general was Leeuwenhoek's microscope, which is what you call a simple microscope. You have one objective and you can place your sample near on the stage and you can see an image of it. And this is this folding microscope is something that tries to recapitulate what Leeuwenhoek did, and you can probably go and look it up as Sir had suggested. So this is So, there are two kinds of microscopes. One of them is a simple microscope in which you have only a eyepiece and a lens, and you see whatever's present depending upon what your zoom of the lens is. What is the so there are different kind of objective lenses: 10x, 20x, 4x. It can go as high as up to 100x. So, but the other kind of microscope that exists is something that is called as a compound microscope, in which you don't have only one lens that would that would zoom out whatever your sample is. You'll have multiple lenses. So instead of there just being an objective, there would be other lenses throughout the light light path that would further uh, zoom the image. Like there would be a lens here which would zoom it up by one x by 2x or 1.5x, and there will be other filters. There will be uh, a diffuser, there will be condenser, all of the works. But that is a compound microscope. When you are thinking about a simple microscope, it's just the objective lens and you see through whatever light passes through this objective lens. Uh, Coming to light microscope, one of the most important concepts in light microscope, and it is one of the most uh, one of the most uh, rep- not reported. It's one of the problems one faces when they are trying to image objects is the diffraction barrier, uh, which is, it should be micrometer, but who knows. So what diffraction barrier is? Simply in air, uh, diffraction barrier means that when the minimum distance that two objects needs to be separated so that you can image those two objects as two separate objects, which is for air medium lambda by 2, which is in which lambda is your wavelength. If you are say passing uh, gr- green light, which is 488 uh, uh, 488 micrometer in wavelength, you would divide it by 2, which is 244, and that needs to be your uh, minimum length of separation between the two objects that you are trying to distinguish. uh, there are other kinds of, this is just a light microscope. Within microscope there are different kinds of microscopes. A bright field is when you try to illuminate the object, and depending upon what light passes through and what does not, you get a black and white image of sorts. Dark field is sort of an opposite of bright field. And it depends on what it generates an image based on what is stopping the light and what is not. um see uh, there are others like polarized light, in which you so light has a certain direction of polarity, right? So the way the light is polarized, randomly, if you just shine bright light like white light onto a sample, that's unpolarized light. If you try to polarize it at a certain angle and then pass it through a object, you would get different kinds of images. Of course, apart from light microscope, there are other methods of imaging samples such as X-ray diffraction or electron microscopy. But we would not be discussing all of this here. Uh, the basic structure of a light microscope is similar to what is there in the eye. You have a lens, you see light passes through the lens of the eye and it hits here at the detector, and you get an image mm, based on the uh, synapse responses at this detector. Uh, in a synthetic light microscope, what you have is an objective which is placed directly over your specimen. There is a tube lens which can either just pass the light that is coming through the objective, or can magnify the image that is coming through the objective. And then finally you have eyepiece. There could be a magnification here at the eyepiece as well, and this the eyepiece is where you place, like, you place your eye beyond the eyepiece, or your detector beyond the eyepiece, and you observe what the image is. um so uh as I said earlier, light can be unpolarized or it can be polarized. If you're using a polarized light, you can get something that is called as a diffraction pattern if you place the light at a certain angle. So it would, what would happen is that there is a layer of, so when you are trying to image your sample, you would your light would encounter different sorts of interfaces. Like, there would be an interface between the air and the, say, specimen. There would be an interface between the objective and air, uh, and all of these would sort of culminate to give you something called as an interference. If you go back to your 11th, 12th, 9th, 10th, whatever standard physics textbooks, you would remember that uh, plain polarized light, when you shine, you get different kinds of patterns depending upon what your shift is in the light polarization. Um, so yeah, this is one of the methods of imaging. It's not light microscopy, but it's uh, interference microscope. Um, now, there are, as I said before, there are different kinds of objectives available. It can be 4x, 10x, 20x, all of 100x, etc. So what happens, as you go higher in the objective zoom, is that you sort of, the interface between air and the objective lens sort of start interfering between your ability to focus on the sample. You can focus on it, but your sample won't be clear. So, as a correction for that, what microscope builders have done is that they have tried to modify the objective lens such that you can place a thin layer of uh, a liquid which is usually has a higher refractive index uh, than air, and you would basically uh, get a better focus using that liquid. So there will, between your objective and your sample, there will be a thin layer of, say, oil or water. So basically these immersion type of objectives come either water immersion or oil immersion usually. And this would basically help you get a better image out of your sample. um, There are, if you go into the specific structure of microscopes, there is something called as a condenser, which, so your light is coming as a big pillar of uh, waves or particles, whatever, and you want it to focus on the specimen that is there. So you don't want extra light on the sides. So what you use is a condenser, in which, if you, so the condenser has different apertures. So depending on what your size of specimen is, you can control the size of the condenser aperture, and this would basically help you get a more clearer picture. And yeah, so I there are, as I said earlier, there are other methods of looking at how what your molecules or samples are, and two of them were X-ray and electron microscopy. Uh, but the thing that we are concerned about right now is light microscope, and since the visible light ranges from say 400 300 to 700, or 250 to 700, you and your minimum, if you factor in the resolution power of the microscopes, you need lambda by 2 if you're considering air. So the minimum, the smallest sample that you can image is 100 nanometers. If you want to go lower, you need to switch to other methods. Um, yeah, these are the different kinds of light microscopes. Uh, Bright field is normal white light passing through the uh, sample. Dark field is the opposite of light field. Phase contrast is when you have uh, uh, different kinds of blockers on the light path so that you get a better image. So uh, and this is what happens in due to diffraction in a polar from a polarized light, etc. um the other a most other famous type of microscopy and that a lot of people use is fluorescence microscopy. So what the concept behind fluorescence is, you shine a particular light of a particular wavelength. Your whatever your molecule you are interested in accepts that light and absorbs some energy, retains it, and then emits a light of a different uh, uh, wavelength. Uh, this is the physics behind it. You can read it up if you want to. But yes, the fluorescence microscopy is super important when you want to observe how your proteins or how specific molecules in your cell are moving around. Um, this is something known as epifluorescence microscope. So your light would pass through here. It would so the fluorescence wavelength could either the light that you generate can be of different sources. It can be a laser, it can be a halogen lamp, etc. So uh, you basically are, because you are shining your light from above and it then reflects from the objective and then passes through your eyepiece. It's called epifluorescence. Epi means above. And um, these are the different kinds of objectives that I was talking about. So some objectives have the capacity. So uh, how thick your sample is also affects how well you can image it. Say, and what your sample is placed on. Say you have a very thick layer of glass between your sample and your objective, or uh, so that would if you are trying to image it from down, from below, uh, if you have a very thick layer of glass in between your objective where your uh, sample is placed on, it would affect your ability to focus on the sample. To obtain an image, you need to place the light rays right to focus on exactly on the layer of the sample that you want. So if you want to if you have a sort of uh, uh, very thick layer, you can use certain objective lenses are equipped with the power to correct for that by changing the uh, size of their um, of the length of their uh, uh, the whole body, and you can basically look it up if you want to, the engineering behind it. These are the fluorophores that uh, are typically used during fluorescence microscopy. So DAPI is something that you use to stain for DNA. It basically goes and intercalates into the double helix of DNA, and uh, this intercalation changes its structure so that when you shine UV light onto this DAPI, onto the sample, you get a blue wavelength out of it. Um, the dill is something that you use to stain for liposomes. So liposomes are basically these vesicles which are covered, which are uh, the, the membrane of which is made up of lipids. There are usually most of the proteins that we use are tagged with a fluorescent protein, extra fluorescent protein. So you would have a protein of interest, and there would be a tag associated with which can be green fluorescent protein, which can be a red fluorescent protein. So the origin of it is from jellyfish, which the name of which I don't remember, but yeah, the basically the structure of GFP protein helps it to the fluorophore that we are interested in is sort of here inside the uh, inside the beta barrel structure. This these beta sheets that surround the fluorophore is what is called as a beta barrel structure. So you basically tag your protein with GFP, and when you shine light, you basically can track where your protein is going. Um, Normal epifluorescence microscope shines light, and it shines light throughout the sample. As and it sort of decreases how good of an image you get. Uh, Subsequent uh, modifications to epifluorescence microscopy led to the birth of confocal microscope. What confocal microscopy is that, suppose you have a sample that is this thick, which is like say 5 microns in thickness. What you want is that you want to focus on one layer of the sample at a time, and that way you can sort of ignore the other layers, the fluorescence coming from the other layers, and this helps you get a more 3D picture of how your sample looks like. um yeah, this basically mentions that. So when you are sort of shining light using a light microscope, what happens is that you would uh, if you want to say detect this layer, you would also detect the layers above and below. But with confocal microscopy, because of how the polarizers and how the filters are present, you only focus on this layer. Now there are two types of confocal microscopy. This is something known as a scanning laser microscope. There is a second type called a spinning disk microscopy, which is slightly so scanning laser microscope is slightly better at obtaining images and at processing it as compared to spinning disk microscope. Um, this is just a light path of the confocal microscope. Yeah, so the larger light that you obtain from your sample, the more diffused your image is. So, and way to what uh, uh, scanning microscope, scanning confocal microscope does is it focus lights one point at a time. So you would first go here and then go here and then go here and then you basically obtain an image of your whole sample. And underside of this is that you would bleach your samples faster. So when you are looking at fluorescent samples, you have a very limited number of fluorophores that are present in your sample, and most of the time these fluorophores, once you excite them and they emit the light, they lose their fluorescence because they can't go back to their ground state. So if you go here, this is the ground state. So after they are emitting the light, they sort of uh, remain here instead of going back to the ground state. And this might not be one cycle, this can be multiple cycles. Also, there exist some molecules which can go back to their ground state very well due to certain chemical chemistry associated with them. However, normally that's a problem with confocal microscopy, that you would basically lose your signal because of bleaching. Um, yeah, it's like a bread loaf, and you image one slice at a time, and you then get a 3D image. So this is not something known as a Z stack. Uh, uh, Z is, so you have the plane at which your sample is placed is X and Y, and the thickness of your sample is basically Z. Um, yeah, these are the processed pretty images that you get from confocal usually. Um, this is the other kind of confocal that I was talking about, in which you, so instead of focusing at one point, it has a disc which here uh, which would pass through certain wavelength, certain rays of light at one point, and but it's not one point at a time; it's multi-point. So you get a lower resolution. This is again the diffraction limit. So there exist other more advanced kinds of microscopy which people have started to call as super resolution microscopy. The super resolution microscopy would, uh, due to the engineering changes to the microscope, they can uh, achieve subrefraction limit, but it's not very uh, it's lower than that, lower than 100 nanometers, but there is it's not as low as electron microscope. One of the examples is TED, in which so instead what happens that is that when you're passing light, you have say this big often beam going through the sample. What STED does is that it places a filter such that the light passing through sort of becomes small. So because you are imaging a smaller area, you get a better resolution. Um, other method, photo switching is something that photo switching is sort of a stochastic activity. Uh, so when you're shining light at very low uh, wavelengths, very low frequencies, what happens is that you only excite some molecules and not all of them. And this sort of helps you to image one molecule at a time, even though they are present very close to each other. Say uh, there are two say there are two samples which are 2 points in your sample which are placed very close to each other under the diffraction limit. The point behind photo switching is that when you shine lights at very low energies, what happens is that you excite this, but you don't excite this in a temporal fashion, in a time dependent manner. And because of that, you only get an image of this and not this. And the next time around, you can get an image of this. So this basic, and then you can computationally sort of club the all of the images that you have obtained and uh, get an image of the whole sample. Yeah, this is the uh, this is basically what I said about the photo switching of the sample. Um, yeah, you can. So, even with all of this, there you would get very grainy results such as this, which you can process further, which is known as deconvolution. And so, if this is your raw image, you process it, you do a Fourier transform, you can look it up, and you get a uh, deconvoluted image basically. It uh, so your light always uh, spreads in a Gaussian fashion, right? So what deconvolution does is it takes into account this much and then it plots this much into your image, and it ignores the spread of the light, the sides of the image, sort of, in very layman terms. Um, I don't think so. Explained on special electron microscopy, but anyways, the basic uh, uh, concept is that you treat your samples with uh, a particular chemical, and these chemicals usually are electron rich, and then you, depending upon how well your sample takes up the chemical, you would have electron rich and electron deprived areas. then you do microscopy and you get image. These are processed images, these are not raw images. The raw images needs to be always in microscopy, the raw images need to be processed for them to get a better image, so that you uh, sort of discard all the junk that came with the image. um One of the other things that is very common is that is cryo-EM, in which you first freeze the sample and then sort of perform electron microscopy in it in slices, so that you can obtain a more 3D uh, uh, 3D view of what your sample looks like. This is basically the biology, the inside of the cell. Okay, moving on. If anyone has any doubts here, I can stop and answer them. Okay. Good. Moving on. Now coming to what your cell looks like and what's present inside the cell. Um, there are two kinds of cells that are present, at least that has been discovered until now. Prokaryotes and eukaryotes. Um, Prokaryotes what uh, are known as the primitive organisms, and eukaryotes are more advanced because they are evolutionarily late. Uh, Evolutionarily, they appeared later than the prokaryotes. Um, so, basic differences between prokaryotes and eukaryotes. Prokaryotes don't have any organelles inside them. They don't have any nuclear bound membrane. They don't have any membranes inside them. The only membrane that's present is the cell membrane. And inside the nucleus is present in an is present bound to the cell membrane uh, in a dense fashion in a region called as the nucleus. In contrast, in eukaryotes, there are various membrane bound organelles present, which leads to compartmentalization of functioning of the cell. And there are also multiple, you also have a nucleus which is membrane bound, and the nuclear contents uh, are sort of placed inside this only. There are no they are not floating around inside the plasma. Um, these are the basic differences. Um, yeah, you have the cytoplasm, you have the DNA. There are the other structures that are present in on the bacteria. Is some of the bacteria have cell wall, which is a peptidoglycan cell wall, and you have uh, uh, you, the motility of bacteria happens through flagella and pili, uh, uh, which basically flagella helps them to move around, and pili helps them to stick to uh, the substrate they want to stick to. Some of the pili is also uh, So, if you remember last week, we had talked about horizontal gene transfer. So there are certain villi in on the bacteria in certain species that which are known as sex villi, which you can transfer your the transfer the genetic material from one bacteria to another bacteria. Um, uh, yeah, that's it. There are other only uh, defined thing present inside the bacteria is ribosomes. The prokaryotes can be of different kinds, unicellular such as amoeba. They can form a colony, what uh, which we which has been studied nowadays known as biofilms, which have certain structural properties which helps them to be of industrial use. They can also be filamentous and form uh, chains of a cell. Some of the examples of colony forming uh, um, feeding style of prokaryotes can either be photosynthetic, in which they have chlorophyll present inside their cytoplasm. They can be disease causing or they can be pathogenic, in which case they feed on living things, or they can be decomposers, in which case they feed on dead leaves or decaying meat, etc. Uh, Eukaryotes have a larger size as compared to prokaryotes. They have membrane-bound organelles. They have a defined nuclear structure. They have chromosomes, chromatids, etc. They can be uh, social, and they can be present in a multicellular fashion, more as compared to prokaryotes. Um, when you think about eukaryotes, you have plant cells, you have animal cells, you have fungal cells. Um, the arrangement in the inside the cell is inside a eukaryotic cell. Given that it has so many different organelles, you would need a very good communication system inside it, and there is a lot of interplay between the components of inside this. There are definitely individual units or organelles present which can move on different uh, if you want to take the subway example, they can move on different tracks, such as the cytoskeleton that's present inside the cell. Um, the individual units also have a defined structure inside them. The uh, and the structure are sort of conserved between different kinds of eukaryotic cells. Um, The cells also have certain communication between these organelles, and there are certain command centers that uh, sort of uh, dictate how the signal flow occurs inside the cell. And this is how you create a cell. There has been uh, a lot of a lot of labs are trying to build artificial cells. From a scientific perspective, you it would get help us get a better sense of how evolution happened, but we'll come to that later. um, I can sort of discuss the leukocyte migration if you want. Okay, can just discuss. Um, so Search showed a video about leukocyte migration. So, your blood RBCs are free floating inside the uh, uh, blood vessel. The lining of your blood vessels is made up of something called as endothelial cell. Now, leukocytes do not free float inside the blood vessels. Rather, they are sort of stuck to the uh, endothelial cell layer, and what happens is that the endothelial cells have P selectins, which is a receptor, which is a protein that is present on the surface of the endothelial cells. Uh, as a counter, the leukocytes have PSGOs on their surface. These PSGOs and P selectins would interact, and the this would cause a rolling motion of the leukocytes. When there is an inflammation, there is a the leukocyte sort of senses the cytokines that are secreted at the inflammation site, and there is a signal processing that happens downstream. This signal processing results in the outflow of the integrins, which are if present inside the cell, it sort of flushes them out to the surface of the cell. And this the integrins, now that they are on the surface of the cell, they can sort of interact with the ICAMs that are present on the surface of the endothelial cells. Now, this results in the leukocyte sort of sticking there, and then it can um, fold itself, it can squeeze itself to go to the inflammation cell. Um, okay. Um, so even in eukaryotic cells, if you're considering a single individual, you there say you are considering humans, there are different kinds of cells present inside one body, and these kinds of cells have specialized functions. Even though the genetic code that encodes for these cells is the same, they can be neuronal, they can be uh, epithelial. In just this is These are the kind of cells that are present in on the intestinal in the intestine. uh, they can be leukocytes, they can be anything. um Now the first eukaryotes showed up around 2 billions of years ago, and the conditions sort of kept changing. There was a drop in CO2 levels, there was an increase in oxygen levels, and there was sunlight, and all of this sort of culminated together to uh, positively select the present day eukaryotes. And now coming to the structures that is present inside the cell. So the cell, the cell is sort of encapsulated inside the cell membrane, which is in case of animal cells, the first barrier that the cell encounters. Now, I had talked about cell wall in case of bacteria. Cell wall is also present in case of fungi and plant cells, but not in case of animal cells. Uh, this cell wall is permeable. It uh, we will be discussing that later on. But say, I the point I want to put across is permeability uh, of a barrier. Cell membrane, on the other hand, is semi-permeable. It only allows certain small ions and water molecules to pass through. Other things that pass through uh, are usually with the help of different proteins or different pores that are present on the surface. Um, the con- constituents of the cell membrane are usually protein and lipids, and these protein and lipids can be modified by different kinds of phosphorylations or glycosylations, and this is found in all kinds of cells, even uh, bacterial cells. The structure of bacterial versus eukaryotic cells might be different though. Um, Now, coming on to the nucleus, which is what stores your genetic material, the nucleus is encapsulated in a bilayer of uh, in a lipid bilayer, and the DNA is sort of folded into chromatids, and these chromatids can then organize themselves to form chromosomes. And the bilayer that surrounds the nucleus has pores in it, and these pores sort of help traffic out the RNA molecules that are created after transcription of the genes in using the RNA polymers. Uh, the other one of the other organelles that is present is the mitochondria of the cell. If you go back to school days, it's the powerhouse of the cell. Um, the mitochondria is has a double lipid bilayer. It it has two layers of bilayers, and it produces it produces ATP in the cell by using electron transport chain. We might be discussing that later on, but otherwise you can go look it up. Depending upon what cell type you are in. So, for example, if you have a muscle cell type, uh, you for muscle cell type, it needs to do a lot of work, so it needs more energy. So that energy needs, because it needs to produce a higher amount of energy, it needs more mitochondria. So if you uh, compare some muscle cells versus intestinal cells, you would see that muscle cells have a higher density of mitochondria as compared to intestinal cells. Um, Also, mitochondria has its own DNA, and we will be discussing that further on. There are ribosomes which are also present in bacterial cells. These ribosomes are responsible for the translation of the RNA into proteins. It can be they can be free floating in the cytoplasm, or they can be associated with the endoplasmic reticulum. Uh, these when the ribosomes are associated with the endoplasmic reticulum, the surface of the endoplasmic reticulum sort of is rougher, so it's called rough endoplasmic reticulum. um, and this is made up of RNA. And now coming to endoplasmic reticulum, this is also membrane. This is also made up of membranes, and as I said before, they can be rough, or if they are devoid of ribosomes, they can be smooth. These rough endoplasmic reticulum are used for storage of proteins and for protein processing. And yeah, that's it. Um, Now, there is uh, another organelle, which is the Golgi complex, which sort of accepts the raw material from ER, such as proteins produced in the ER, and then it processes those proteins. It imparts modifications to those proteins if they need to be phosphorylated, if they need to be uh, ubiquitinated. It does all of that, and then it sends the uh, proteins out into the cytoplasm. Or if these proteins are transmembrane uh, proteins, then the- they are sort of poked into the Golgi complex's membrane, and uh, the, the area of membrane which contains these proteins bursts off from the Golgi complex and goes onto the membrane. It can be cell membrane, or it can be ER, or it can be mitochondria, and it attaches there and uh, performs its function. Um, the other membrane bound organelle is lysosomes. These lysosomes are responsible for keeping the cell clean, so to say. These are filled with digestive enzymes, which, if present in the cytoplasm, could destroy the cell. Uh, they take up the worn out, used up parts of the cell and digest it, and then recycle these parts and give it back to the cell for reuse, or they throw it out of the cell if needed. Centrioles are, so we had talked about the tracts, which are cytoskeletal, uh, which is the cytoskeleton of the cell. Now, the cytoskeleton of the cell contains multiple uh, multiple uh, components inside it. They can have microtubules on which the cargo usually. When I say cargo, it's the thing that needs to be transported in the point A of the cell to point B of the cell. It can help in keeping uh, the uh, a particular organelle fixed at a point, but I think that will be discussed next week. There can be actin filaments which sort of help in migration and squeezing and uh, whatnot. And so the microtubules that uh, help in cargo trafficking are uh, originate from the centrioles. These centrioles are usually present near the nucleus, and are the microtubule organizing center. Um, so they are also responsible for division of the cell, when the cell is ready for dividing, when it goes through the certain cycle of division. And next is the cytoskeleton, which is microtubules, actin filaments, intermediary filaments, etc. It gives the cell shape, rigidity, support, and it also keeps the organelles in their place. It also helps the cell move around if it needs to, like in case of um, leukocyte migration. If the cell had to, at the end point, needed to squeeze through the endothelial layer to reach the inflammation. The squeezing and moving is the work of the cytoskeleton, particularly actin filaments. Uh, Now, apart from these basic uh, organelles, there are other parts of the cell. Cell wall is one of them, and in case of plant cells, it's usually made up of the major component is cellulose. In case of fungal cells, a major component is chitin. This basically helps in protecting the cell from rupturing and provides the stability and uh, forms uh, junctions with the other cells. Uh, this lets all of the chemicals or all of the molecules to pass through. Um, the only barrier that stops them from entering the cell is the cell membrane. Vacuole is a huge filled sac inside the cell. It can be water, it can be, there are vacuoles which are rich in certain components, it can be starch, it can be lipids, etc. It uh, if it's filled with water, it keeps the target pressure, which is the pressure inside the cell, and keeps it rich. Um, then there are chloroplasts, which are filled with chlorophyll and help to do photosynthesis. These are something that's specific to the plants. Now, coming to the complexity of uh, uh, the cells, and why there was a need of compartmentalization. So, prokaryotic cells are very small in nature, and they are, given that they are small, they the dynamics of moving the things from point A to point B in prokaryotic cells is very easy. They can, in simple cases, they can just diffuse. However, in case of eukaryotic cells, since they are mm, in the order of micrometers, multiple micrometers, it's very difficult for things to diffuse from one place to another, and uh, there and um, if you have made something in point B, A, and it needs to go throughout the cell, it and if there are multiple points A in the cell, if there is a one organelle which is present at multiple points, it makes the cell very crowded. So, compartmentalization was sort of a necessity to make the cell more efficient in doing what it does, and uh, it sort of produced a division of labor so that every organelle uh, has a fixed function associated with it, and there is no mismatch, and this basically also increases efficiency. um, One more thing is that there are downsides to this compartmentalization. You need coordination between all of the organelles that are present inside the cell. The nucleus needs to make the RNA at a certain point. This RNA needs to go to the cytoplasm at a certain point and needs to be translated in the ribosomes at a certain point. So there needs to be a lot of networking that is present inside the cells for it to function efficiently. Um, Different genes expressed in the cell would result in different kinds of cells. So the genes that are expressed in, say, epithelial cells would not be the same genes that are expressed in the neuronal cells. Okay, now this is the last one. Um, a major question that has been prevailing in the biological community is where the how did the complex eukaryotic structure arise? The prokaryotes just had a nucleoid and they were but the conditions changed and the prokaryotes modified and they formed proeukaryotes. But where did these extra organelles come from? One of the famous theories is the endosymbiont theory, which yeah, which they essentially means that endo means the word break is endo means inside and symbiotic means friend. Uh, the organism that was engulfed reaped certain benefits from the host cell, and the host cell reaped certain benefits from the engulfed organism. Now coming back, okay. So the theory goes like this: is that uh, there was a proto eukaryote, and it engulfed the mitochondria. So therefore, the mitochondria was initially a prokaryote, which was aerobic in nature, and it engulfed in it inside a membrane compartment. So therefore, it's a double membrane. Then, as a subsequent step, this there was a there was a diversification in which certain of these mitochondria containing cells engulfed photosynthetic bacterium, which led to the production of chloroplast inside the eukaryote, and some didn't, and these were heterotrophic in nature. Um, Lynn Margulis is the person who has popularized the endosymbiont theory, and there has been a lot of work. They have tried to sequence the so both mitochondria and chloroplast have their own DNA associated with it, which is different from the DNA that's present from in the nucleus of that host cell. So there have been multiple studies in which they have tried to compare uh, how the sequence of the DNA varies in case of mitochondria, chloroplast, and prokaryotes, and um, they see that the mitochondrial DNA and the chloroplast DNA is similar to what is found in other prokaryotes, even at the same present day. This is another depiction which explains how the mitochondria and chloroplast came to be in the cell. And yeah, uh, there a lot of studies have tried to map out exactly what the ancestor was whose engulfing led to the production of the first eukaryotic cell, but it's still ongoing. Um, other except for the comparison of genetic sequences, uh, other leads were in case of the in case of the amino acids codons. So in case of normal universal codon anticodon concept, you see that UG goes for stop when, whereas in case of the human mitochondria it codes for tryptophan. Uh, Whereas AG, which is supposed to AG and AGG, which are supposed to code for arginine, code for stop in case of mitochondrial growth. Similarly, AUA, which is supposed to code for isoleucine in case of normal cytoplasmic ribosomes, it codes for methionine in case of mitochondria. So these are sort of reminiscent of what how the bacterial bacterial codon looks like. So this combined with sequence data led to a belief that the mitochondria and chloroplast have sort of emerged from different prokaryotes, and uh, therefore the endosymbiont theory came into being. This is another depiction of hypothesis of how the engulfment may have been. Yeah, that's it. If there are any questions, I can answer now, or I can discuss the assignment questions. So let me discuss the assignment question first. Okay. Um. So, I the first question is that which of the following statements about endosymbiont theory is true? Mitochondria evolved from photosynthetic bacteria while chloroplasts were aerobic bacteria. It's strong because mitochondria is evolved from aerobic bacteria, which is why it has the ability to perform the electron transport chain and all that. And chloroplast is evolved from photosynthetic bacteria and therefore has chlorophyll present inside it and can perform the function of photosynthesis. The other options are also incorrect. Which of the following is not a difference between prokaryote and eukaryote? Size, it's different. Prokaryotes have a size of 1 to 10 microns, whereas eukaryotes can go up to hundreds of microns. Organization of DNA is again different. Prokaryotes have nucleoid, whereas eukaryotes have nuclear membrane bound nucleus present inside. Uh, However, both of them use ATP as an energy source. Mitochondria again is not present in prokaryotes, whereas it is present in eukaryotes. Presolving power is the ability of microscope to distinguish between two adjacent objects in the distinct as distinct. Focus is when you try to shine your light at a particular place inside your sample, and how the light rays are arranged defines where your focus is. Wavelength is the wavelength pertains to the uh, light that you are shining on your sample, and if it's a single uh, wavelength light, it can have uh, and belongs to visible spectrum. It can have anything between 300 to 700 na- nanometers. Magnification is again associated with uh, objective lenses and well what how well they can magnify the sample that you are trying to observe. That is the ability of molecule to absorb light in one wavelength and emit the same or longer wavelength. Now, chemiluminescence is not. Chemiluminescence is more related to the mixing of certain uh, reagents, and it gives you a new compound which is chemiluminescent in nature, which is not what we are interested in. Spontaneous emission and radioactivity are also just do not explain this particular statement. Fluorescence is the ability of the molecule to absorb light and then emit it. Usually, it's not the case that the light that is emitted is of the same wavelength. Usually, it's of a longer wavelength because some of the energy is retained, and physics, you longer the wavelength, lower the energy. Um, which of the following cell types contain plasma membrane? All of the cell types, plant, animal, bacterial, fungal, all of them have plasma membrane. Uh, the typical size of a prokaryotic cell is in the range of 1 to 10 microns. Uh, which of the following statements is true? Eukaryotic cells contain DNA, whereas prokaryotic cells lack DNA, which is not true. It's just that the organization of DNA is different. Both prokaryotic and eukaryotic cells contain nucleus bound by nucleus membrane, which is not true because prokaryotes are devoid of nuclear membrane. Prokaryotes cells lack membrane-bound organelles, but prokaryotic cells possess membrane-bound organelles, which is true. Prokaryotes have a cell wall made of chitin, and eukaryotes have a cell wall made of peptidoglycans. This is also false. Prokaryotes have a cell wall made up of peptidoglycans, and in eukaryotes, the fungal cells have cell walls made up of chitin. The plant cells have cell wall made up of cellulose. Uh, all of these are different complex carbohydrates. Um, which of the statements on the organization of DNA in prokaryotes and eukaryotes is false? In eukaryotes, the genomic DNA is packaged into chromosomes, which is true. Prokaryotes carry a single double-stranded DNA, which is true. Same goes with mitochondria and chloroplast. In eukaryotes, the genomic DNA is condensed and held in a region called nuclear. Yes, the genomic DNA is condensed, but it's not in nuclear, it's in nucleus. Um, in prokaryotes, the genomic DNA is not separated from the rest of the cell by a nuclear membrane, which is also true. Which of the following features supports the idea of mitochondria being an endosymbionts? Mitochondria is double membrane bound organelles. As I mentioned before, the when the organism was being engulfed by a larger cell, uh, sort of engulfed in a pouch of membrane. So it the organism had its own membrane, and it had a membrane outside which belonged to the parents or belonged to the host cell. Mitochondria carries its own DNA, which is single, uh, which is circular DNA. There are examples of similar codons for different amino acids in mitochondria versus nucleus, and which is true. And these different amino acids also are present in the bacteria. So all of these are true. What are the two key differences between plant and animal cells? Presence of cell wall and chloroplast in plant cells. True. Vacuoles are also present in plant cells and not in animal cells. Ribosomes are present in both plant and animal cells. Uh, yeah, and mitochondria is also present in both plant and animal cells. Uh, yes, that is it. Does anyone have any questions? Or they want me to explain something else? I can hang around for a few minutes in case any of you have any questions. Otherwise, this is the end of this session.